Yes, melanocyte, we have melanocyte transporting substances. For example, tyrosine into the melanocyte. We have enzyme. We have substances within that melanocyte. If one of the transporting molecules transport substances into the melanocyte so that it can produce melanin is defective, the gene producing it is defective, then somebody can still get albinism. So it is not about a lack of enzyme alone, it's into the melanocyte where now they can be used to make melanin. So you're saying the inability of enzyme to enter the pigment. This enzyme does not just enter the pigment, it has to be transported by a transport protein. So a transport protein, a gene coding for that transport protein can be defective. And so again, somebody will not be able to produce melanin because substances that are needed inside the melanocyte have not been taken into because you lack a protein responsible for transportation. So I'm saying the inability of an enzyme to enter the pigment cell and convert amino acid tyrosine to melanin. So those people, all those. So when we talk about albinism, we are talking about a person must have the two recessive alleles or the genes. Another disease that is inherited recessively, meaning you have to have the two defective genes for you to get the disease, is something called phenylketonuria. Phenylketonuria is a disease that affects a breakdown of phenylalanine to actually, when you're breaking down phenylalanine, you usually produce tyrosine. The enzyme that is responsible is called phenylalanine hydrolase. So the gene producing that phenylalanine hydrolase, you must be defective. You must have a recessive gene for that phenylalanine hydrolase. So what happens if you're not able to break down phenylalanine? Phenylalanine will actually accumulate and when it accumulates, it leads to this disease called. So you're saying recessive because other large neutral amino acids, which are usually transported by a transporter across the blood-brain barrier, will actually fail to be transported, yet they are needed by the brain to make important neurotransmitters. For example, tyrosine is used to make what? Dopamine. So if this accumulation of phenylalanine will saturate the transporter because they all share one transporter. It will saturate the transporter. Other amino acids such as dopamine, such as tyrosine, will not be able to use that transporter. And so important neurotransmitters which are made from these, which are needed by the brain, will not be produced because other amino acids will be blocked from being transported due to accumulation of this phenylalanine. Another disease that is inherited recessively is Tay-Sachs disease. So when you talk about recessively, I've told you must have the two recessive alleles being defective. So another disease is called this Tay-Sachs disease, and there is what fats. Actually, it is a lipid storage disease. So fats will not be broken down. One of the fat that is accumulating in the nervous system is the sphingomyelin or sphingocytes. So what happens? They are not broken down. And so they accumulate in the nervous system. And when they accumulate, they lead to degeneration of the nervous system. So you say it's an autosomal recessive resulting in degeneration of the nervous system. So the same symptoms manifest after birth, and child homozygous recessive for the allele rarely survive past 5 years. So sufferers lack the ability to make an enzyme called N-acetyl exosaminidase, which break down GM2 ganglioside lipid. GM2 ganglioside is an example of a sphingolipid. So this lipid accumulates in the lysosomes in the brain, eventually killing the brain cells because they need to be broken down. But now when they are not broken down, they accumulate and lead to that disease. Sickle cell anemia, I think we talked a little bit last time about sickle cell anemia, but it is one of the diseases that is inherited recessively. For somebody to manifest the disease, you must have the two defective genes. And how does sickle cell anemia occur? So you're saying it is an autosomal recessive with beta genes of hemoglobin. That becomes a T. When during transcription, it becomes adenine, becomes a thymine. It becomes what? The amino acid that will be produced from translation will be glutamine, and this other one will be a valine. So when there is a valine instead of glutamine, automatically that protein is affected
and that is what is responsible for development of sickle cell anemia. So so we're saying when oxygen concentration is low, usually because the protein is not formed properly, it causes sickling of cells to occur. So abnormal red blood cells usually, so when sickling of cells occur, there is two effects. One, these sickle cells will be removed by the immune system because they look abnormal. So abnormal red blood cells are usually removed, resulting in anemia. The other thing is that these sickle-shaped red blood cells tend to stick in the capillaries. And when they stick in the capillaries, they will deprive tissues of oxygen and they will lead to cell death. So this is now the manifestation of sickle cell anemia. And most of the time, they usually sickle when there is low oxygen concentration. So the consequences is that it can lead to organ failure. Why? Because when the cells sickle, they get trapped in the capillaries, and so they don't transport oxygen to the required organs, and so there is organ failure. So you're saying heterozygotes make enough good beta chain hemoglobin that they do not suffer as long as oxygen concentration remain high, such as at sea level. So those who are heterozygotes behave normally, especially at when they live where there is enough oxygen concentration, for example, living at above sea level. Now, another disease that is inherited again recessively is a disease called cystic fibrosis. So cystic fibrosis is whereby somebody has cysts, especially in the lung, due to a defect of something called cleon transport. So it is inherited recessively, and you're saying heterozygous carriers may be less likely to die from, let's say from diarrhea-inducing diseases. But most of these cysts usually occur in the lungs, and it is due to that defect, and it is inherited recessively. Now, we have human allelic disorders that is dominant. So when you talk about dominant, you must have a dominant allele for it to manifest this disease. So, for example, if these are carrier parents, so the chances of getting a child who are sick is usually 50% chance of being affected by this condition, and it is inherited by a dominant, and you must have the dominant allele for you to get the disease. So one of the diseases is called Huntington's disease. It is an autosomal dominant disease resulting in progressive destruction of brain cells. So if a parent has the disease, 50% of the children will have it. The disease usually does not manifest until after 30 years, although some instances, early onset of this disease are usually reported among individuals in their 20s. Another disease is not a disease, actually, it is a condition we have polydactyly. You know, polydactyly is a condition whereby somebody has an extra digit. It has an extra 6 digit. So in modern times, the extra digit can be cut off at birth, but it is inherited when somebody gets a dominant allele. So when it is expressed using a dominant allele, it's not a must that you have the 2 recessive gene to get the disease. You must have at least a dominant allele for you to get the disease. We have muscular dystrophy. Is a term. This one is involved muscle wasting. So one of these diseases is the Duchenne muscular dystrophy, which affects cardiac, skeletal, as well as some mental function. So DMD is an X-linked recessive disorder which occurs in 1 in 3,500 newborns. Most are before their 20th birthday. Now, we have X-linked recessive inheritance. So when you talk about X-linked, we are talking about diseases that are inherited recessively and are found on the X chromosome. So some of the features is that an X-linked recessive trait is mostly seen in males, and it is uncommon in females. Why? Because man has only one X chromosome. So the trait is transmitted from an affected man, or through all his daughters, because if at all the man has that recessive trait in the X chromosome, and automatically he will give that X to the daughter, that disease will be transmitted to his daughter. So the trait can be transmitted through a series of carrier females. So example of this X-linked recessive inheritance are hemophilia, color blindness, the same Duchenne, muscular dystrophy, and glucose 6-phosphate dehydrogenase deficiency. So hemophilia is a disorder. It is characterized by deficiency of blood clotting protein that results in abnormal bleeding. So there are 3 types, A, B, and C. Hemophilia A is a genetic disorder characterized by deficiency of factor VIII in the blood, which is synthesized mainly in the liver, and is one of the many factors involved in blood coagulation. Factor VIII gene is usually located on the X chromosome. That's why in case you have that mutation or that in that X chromosome, then automatically you end up getting hemophilia. So since males have only a single copy of most of these genes located on the X chromosome, they cannot offset the damage to that gene with an additional copy as the female. So for the female, they are lucky because they have another X. So in case there is mutation in that X, then the other X will still mask the mutation. So consequently, X-linked disorders such as hemophilia are far more common in males. So type A is the most common one, is the common. So 1 in 10,000 males is born with the deficiency or dysfunction of factor VIII. Hemophilia B is caused by deficiency of factor IX and is known as Christmas disease. It occurs in 1 in 100,000 male births. Now we have colorblind, is another X-linked disease. So it has it is a disease that is associated with inability to see various colors or distinguish colors. So it came into limelight in 1798, whereby a first paper was published by this person called Dalton, and he's called it Extraordinary Facts Relating to Vision Colors after realization of his own color blindness. So because of his work, the condition is sometimes called Daltonism, because he was called Dalton. So the most common type of color blindness is red-green hereditary genetic photoreceptor disorders. We have Duchenne. I think we had mentioned about it. It's result from mutation in the dystrophin gene on the X chromosome. So the most distinctive feature of this disease is progressive proximal muscular dystrophy. So this is how it looks like. So this is somebody who has, these are 2 biceps. Somebody who has Duchenne looks like this. The biceps are not well formed. So the most, we are saying there is progressive proximal muscular dystrophy with characteristic pseudo-hypertrophy of calves. So on average, a typical patient with DMD is diagnosed around at the age of 5, is wheelchair dependent at 12 years of age, and is dead before the age of 20. So 1/3 of cases represent new mutations. So there is new mutation continuously, and 1/3 of these cases represent new mutations. Now let's look at Y-linked inheritance. I hope we are together in class, so we can proceed. So when you talk about Y-linked mutation, you're talking about mutations that are linked to the Y chromosomes. So males alone get affected again, and affected males transmit the trait to all his sons because they are the only carriers of the Y chromosome. So the Y chromosome is single. Genes on the Y chromosome are always expressed phenotypically. Example of this Y-linked inheritance are hypertrichosis penile, that is presence of hair in the ears. We have azoospermia, which is complete inability to produce sperms. We have abnormal or absent it's called testicular development, that is gonadal dysgenesis, which is caused by deletion of the SRY gene. We have retinitis pigmentosa, which is an eye disease caused by mutation in the RPY gene. All these genes are located in the Y chromosome. So symptoms of this disease, of this eye disease, is progressive loss of sight, starting with decreased night vision, followed by loss of peripheral or side vision, and finally blindness. Let's look at a small thing here called DNA polymorphism. So DNA polymorphism is what? This is when there is changes in DNA sequence. So poly means many, and morphis means the form of that DNA. So polymorphism, when you talk about genetic polymorphism, is that there are different forms of DNA sequences. So one of them can be a single nucleotide polymorphism, whereby that gene, for example, C cocytosine changes to a thiamine. So a single nucleotide polymorphism is a sequence of DNA on which humans vary by one and only one nucleotide. So because humans differ by one nucleotide per every 1,000 or so nucleotides, there are million SNPs scattered throughout the human genome. So the major advantage of this lies in the fact that they can be detected easily using some DNA arrays, some techniques, some specialized DNA techniques which can detect this polymorphism. So an example of apart from SNP, we have tandem repeat polymorphism, whereby there is repeat of nucleotides. For example, you can see GCAA is repeating in the first diagram, it is repeating 4 times. In the second diagram, it is repeating 5 times and 8 times. So these are tandem repeats. And this tandem repeat can be classified as either microsatellites or mini satellites. So microsatellites is when the repeat is short. For example, it can be 5 to, okay, the other one you were looking, they were 4 to 5 to 6 repeats. So when it is less number of repeats, we call it microsatellites. So the number of repeats is small, usually 5 to 6 or fewer. Mini satellites, the number of repeats is usually large, particularly when it is located in the telomere. So we have something called structural variants. This one is whereby the structure changes, maybe through deletion. It's also a form of polymorphism, or insertion or inversion. So when you talk about structural variant, a structural variant refers to spelling variation that involve deletion. Spelling, we are talking about spelling in terms of bases that involve deletion or insertion of nucleotide sequence, inversion or translocation. So when the structural variant is somewhat large, for example, sometimes genetics define large as 1 kilobase or more, others 10 kilobase. The polymorphism is called a copy number variant or CNV. So this is an example of CNV. When it is large, when the insertions and causes that polymorphism, when the insertion or insertion become large to something like a kilobase, for example, then we call that insertion what? CNV or a copy number variant. So I think that marks the end of the class today. So we have that diagram. We are saying it results from mutation in that gene called dystrophin gene. Dystrophin is the longest, it's a protein. It's the longest protein. It's the longest gene. It's the largest gene in the body, actually, that makes the muscles either to relax and to contract. So we are saying it causes, when you have that mutation, the dystrophin protein, which is responsible for making the muscles, there is progressive proximal muscular. So we're saying it causes progressive muscular dystrophy with characteristics whereby instead of the curves forming properly in the muscle, you can see from the diagram, the muscles are not actually forming properly. So the patient end up having weak muscles ranging from the heart muscles, all the muscles that makes the body. Now this is an example of a biceps. So what will happen? The child is generally weak and so cannot be able even to stand. That's why we are saying a child will be wheelchair dependent even at 12 years of age.